Hello. Today we're going to look at the introduction to cells. So first we need to look at what a eukaryotic cell is. So here we have a cell. That's the membrane, and this is the nucleus. And these are organelles. So a eukaryotic cell is a cell with a true nucleus, a nucleus bound by a membrane, with other membrane-bound organelles. So, for example, in a prokaryotic cell, the DNA will be floating everywhere in the cell, whereas in this case, in the eukaryotic cell, all the DNA is combined, is collected together in a membrane. So that's a eukaryotic cell. Now we'll look at the different organelles of a eukaryotic cell. The first one we'll look at, drawing our cell again, is the cytoplasm. So here we have the membrane, here we have the nucleus, and this gap in between is the cytoplasm. And the cytoplasm is important because that's the space where the reactions occur and the organelles can perform their jobs. And the actual substance is called cytosol. So we have cytosol. So the space between it, we call it the cytoplasm, but the actual substance in it is called cytosol. Now we'll look at the important organelles, starting with the endoplasmic reticulum. ER. Actually, before that, let's focus on the membrane. So the membrane is a lipid bilayer with proteins. What does that mean? Well, this is what the membrane looks at in a miniature view. Looks like. We have hydrophilic, water-loving polar heads, and we have hydrophobic, water-hating tails. Now, this is the extracellular fluid outside the cell, and this is the intracellular fluid inside the cell. And what this means is that this is a lipid bilayer. Bilayer meaning two, and lipid meaning insoluble in water, but soluble in non-polar solvents. I'll cut to the chase. This is a membrane. The circles love water, the tails hate water. Because the tails hate water, they actually come together and form this barrier where things can't pass through. The reason why things can't pass through is because let's say I have a sodium ion. This will be bonded to other water molecules, like so. Now, because the water molecules are attached to it, the water molecules can't pass through. Remember, hydrophobic. So thus the sodium cannot pass through. So it acts as an effective barrier with semi-permeability. Some things can pass through, like oxygen and carbon dioxide, but most things can't. And within the membrane are proteins as well. We have integral proteins, which go all the way through the membrane, and integral proteins may also be channel proteins that allow things to pass through. Or they can be peripheral proteins, where they partly interfere with the membrane, like maybe at the top here or at the bottom. And on top of the membrane, we have carbohydrates as well. So carbohydrate may be here, or carbohydrate may be attached to a protein. These become glycolipids, and these become glycoproteins. Glyco meaning sugar. Now, the term amphipathic. Also, these lipids are called amphipathic because they have a hydrophobic and hydrophilic section. Next, also the carbohydrates have many functions. Most of them are negatively charged, so it makes a cell negatively charged and thus repel other objects. But also, they can bind to other carbohydrates, so they can attach to other cells and form tissues as well. They act as receptors for binding hormones, and also in the identifiers in immune reactions. Next is the endoplasmic reticulum. The ER. So we have the So, before that, a ribosome makes proteins, and you have some free-floating ribosomes. They make proteins for inside the cell. But the endoplasmic reticulum, so this is the nucleus. The endoplasmic reticulum is right outside it, like a maze runner kind of thing. We'll get to that later. These ribosomes attach to the endoplasmic reticulum. They make proteins for outside the cell, in the body. That'll be important later. So, what does the endoplasmic reticulum do? The endoplasmic reticulum gets the made proteins, folds them and processes them, makes them adequate to be used, and then sends them out ready to be transported throughout the body. So, drawing this bigger diagram. This is the nucleus. And here we have the endoplasmic reticulum connected and everything. Now, first we have the rough endoplasmic reticulum. The reason why it's rough is because there are attached ribosomes to it, such as the dots here. These are parallel flattened membrane sacs called cisternae. So it's like a maze it's like a flat, straight maze that you find on a piece of paper. That's what it's like. Then we have the smooth endoplasmic reticulum, kind of in circles. What's happening here is that it's a network of tubules, To draw it vertically, it'll be like this kind of thing, like a network of tubes. And it has no ribosomes. That's why it's smooth. And the purpose of this is to synthesize other important ingredients in cell functions, such as the synthesis of lipid substances, which go to the membrane, as well as detoxification of drugs. Also, with the endoplasmic reticulum, once a protein is made and folded and processed, it gets sent to the Golgi apparatus in vesicles. We'll get to that now. So, now we have the Golgi apparatus. The Golgi apparatus is a secretory organelle, meaning its job is to release things. It releases things within the body, and it releases things outside the cell as well. So the cisternae in the endoplasmic reticulum, a cisternae gets parts of it get binded off the ER and forms this endoplasmic reticulum. It's a collection of flattened vesicles. This is the cis side, meaning the entry, and this is the trans side, meaning the exit. So this is the direction of production. So proteins from the rough endoplasmic reticulum, so this is the RER, will travel to the Golgi apparatus. And the Golgi apparatus will modify, sort, and package these proteins. For example, if the rough endoplasmic reticulum makes a protein important for the blood, the Golgi will understand that and put it in a vesicle that leads to the membrane. If it makes an integral protein for the membrane, the Golgi will understand that and send it to the membrane. If it makes a hydrolase enzyme for lysosomes, it'll understand that and it'll make a lysosome with those enzymes within the cell. So it understands what it's getting and it's putting it in the right place. It's like a librarian. It gets the books and it puts it in the right place within the bookshelf. Next we have lysosomes. Lysosomes are spherical membrane organelles containing hydrolase enzymes. So we have hydrolase. These are powerful enzymes that can break down lots of substances. And basically the point is that they are the digestive power of the cell. They engulf aged or broken organelles, they engulf waste, and they destroy it so it doesn't hurt anything. So their function is digesting particles taken in by endocytosis, degrading old non-functional organelles, performing metabolic functions, and performing apoptosis or cell death. Next we have peroxisomes, very similar. Peroxisomes are similar to lysosomes. However, they are very powerful enzymes that do oxidative reactions rather than broad digestion. They're the more specialized, more powerful lysosomes. So peroxisomes, they produce and neutralize hydrogen peroxide. Hydrogen peroxide is very important in reactions, but too much of it is very dangerous. So they produce it and neutralize it depending on the amount in the cell. They also detoxify harmful substances, such as fatty acids, amino acids, and toxins. And I'm pretty sure they also do peroxisomes neutralize free radicals. Yes, they also neutralize free radicals as well. Next we have the nucleus. And then we'll get to the mitochondria. We have the nucleus. So with the nucleus is membrane-bound organelles that house genetic material. So here is the nucleus, and here we have the nuclear envelope, the nuclear membrane, in other words. And this nuclear envelope has two membranes in it, similar to the bilayer membrane. It has the outer membrane and the inner membrane. The outer membrane connects with the rough endoplasmic reticulum, as we mentioned before, and the inner membrane has a protein network, otherwise the cytoskeleton, for support. So these two combined keep it in that same shape and keep it very structured together. Next we have nucleolus or nuclei, plural. This is the center of the nucleus. That's where the dense non-membrane subregion. No, it's very dense, but it doesn't have a membrane. It's just very connected together. And is the site of rRNA synthesis and ribosome activity. Finally, we have the space in between, the chromatin. The chromatin is a collection of DNA and histone proteins. And there's two sections to this. We have the euchromatin, which is loosely packed active regions where genes are being transcribed. And then we have the heterochromatin, which is tightly packed inactive regions. So the chromatin is either doing something or it's not. Now we have the mitochondria. The mitochondria are very important. Mitochondria allow cells to extract enough energy from nutrients and perform cellular functions. So when we get nutrients, organelles can't just turn into energy. It needs to be broken down and reacted with to get energy, ATP. So that is why we have mitochondria. Mitochondria break down the nutrients, break down the glucose to form ATP and other reactants. And the mitochondria is very, very interesting. It has a outer membrane and an inner membrane as well. So the outer membrane has a lot of channel proteins or porins that regulate the flow of ions and metabolites across the membrane. So everything coming in and out of the mitochondria goes through these holes, these pores. Next we have the inner membrane, or the cristae. The cristae is folds of the inner membrane. So the inner membrane is folded to maximize surface area. So the cristae is folded, and that provides a large surface area for chemical reactions. And inside the inner membrane is the matrix. And it contains enzymes that oxidize nutrients. And that produces ATP, adenosine triphosphate.